Hello everyone. So once again I welcome you all to my series of lecture, that is Understanding Pharmaceutical Science with Dr. Hariharan. So today we are going to discuss about the production of smallpox vaccine. So actually whenever I tell the smallpox vaccine is one of the important vaccination which has been lead to the development of the entire concept of immunology itself. So in this lecture we are going to discuss about the smallpox vaccine, its normal production process. Then we will discuss about the production of smallpox vaccine from the fertile egg. So finally we will discuss about the dosage and the storage condition. So first we understand about the smallpox vaccine. So whenever I talk about vaccine, the person we will come into our mind is the Edward Jenner. He was an English physician, so who has developed the vaccination technology, the idea of vaccination. So what actually he has done is he has taken a small liquid portion of the cowpox virus and he has given administered to the children of the milkmaid, and later on he found that the children were immune to the smallpox. He find these both viruses, that is the cowpox as well as the smallpox viruses, are closely related enough so that it has produced an immunization level to the person. So why this idea of this Edward Jenner is came out, it is the virus which infects humans can cause us toxicity to humans, whereas it doesn't produce any effects on other species. Similarly, the cowpox is a virus generally infects on cattle. It never causes that much infectious disease to the human. So he has chosen this cowpox as an alternate to the smallpox virus, and he administered it and he got the result. So this was the point where the smallpox vaccine development has been initiated. So if you see that the smallpox vaccine is naturally made from this cowpox strain nowadays also. So that is the variola virus. This virus has been attenuated by passing through a series of passages through animals such as cows or rabbits, and this has been used nowadays for the production of the smallpox vaccine. So if you see the concept of production of smallpox vaccine, the smallpox vaccine has been produced from two sources, one from the animal source and second is from the fertile egg source. So in this lecture we will understand both the sources, how the production is done from the animal source and how it is done from the fertile egg. So first we understand the production of smallpox vaccine through an animals, so which is the earlier method. So this production is done by the series of some 5 steps. So if you consider, the first one is that the selection of animal which is used for the production. Second is the immunization of the animal, that is the inoculation with an antigen. And third is incubating until it produces the enough amount of the virus particle. And fourth is the harvesting of this virus particle, smallpox virus particle or the cowpox virus which is used for vaccine. And next is the 5 is the purification process. So it has been purified in such a manner that it without any effect it can be easily administered to the patient. And if you see one by one, in the first step, the selection of animals. So we can use different animals, maybe a calf, rabbits or sheep. So generally preferred is the healthy calves or sheep can be used for the production of the smallpox vaccine. So once we select these animals, we will quarantine it until we see that it doesn't have any communicable diseases. It is examined thoroughly that no communicable disease will exist. Later on it is used for the inoculation of the antigen. So what we do is we select either the flanks or the abdomen. So generally in the case of sheep, the flank is selected because the abdomen is not easy to clean. That's why we select it. Then with the help of a scrubber, we scrub the place, disinfect it, shaved it, then we will re-scrub it and we will re-disinfect it. So while scrubbing is done with the help of a comb which is carrying nail-like portions. While we scrub it, it doesn't draw the bloods. It only produces a scratch-like condition to the animal. Then we will rub this with a seed viral culture of known potency into the scratches. So sometimes the seed can get propagated in rabbits, it is known as the lapine. So generally the seed will start developing the viral infection. So we will incubate it, the animal. So during the incubation, we have to take care. Generally the incubation is done for 4 to 5 days. So until the virus will develop during the line of scarification, and throughout this period the precautions should be taken, because since we scratch the area, it is prone to get infected with other bacteria as well as other virus. So once we scratch it, this area should be aseptically clean and maintained. Only the smallpox virus can grow in that area. So after enough incubation period, enough the viral count is increased into the animals, the next step is the harvesting. So the animals are sacrificed and it is exsanguinated and washed. So after it is exsanguinated, washed, the area which need for the isolation of the virus, variola virus, is the lymph. So the contents of the lymph is removed by curettage, then it is pooled, then homogenized with an homogenizer. So after this, it is pulled and homogenized, it is kept separately. It can be used for the further process until a post-mortem is done to the animal and it is checked whether it has been infected with other bacteria or virus. So it doesn't have any diseases, it is confined. Later on, this material is get purified to isolate the variola virus which is used for the vaccination. So the purification is done. So it is generally when we make a scar, it is very difficult to completely prevent from the extraneous microorganisms. So there is a chance of infection is always possible. So the lymph material which is collected and homogenized is generally treated with killed pathogens. What happens by adding this killed pathogens, the antibodies which is produced against that bacteria might be infected, it gets neutralized and the count of the bacteria gets gradually reduced to a low level. Then what happens is, then we do purification by different methods. So if you see the methods of purification in the older days, the purification is done by grinding with a equal volume of glycerin, and it is stored for a very long time at minus 10 degrees Celsius. So nowadays this purification method has been modified. If you see the modern method of purification, so the lymph material is generally treated with a protein solvent such as trichlorofluoromethane. So this will reduce the lowers the efficiency of the bactericidal agent. So this material is added, so it removes any antibodies against something is present. Then phenol is added to this solution. So around 0.4% of phenol is added, then it is incubated at 20 degrees Celsius for 2 days. So during this time the bacterial count will drastically get reduced, because phenol doesn't affect the virus, but it has a profound effect on the bacteria because it is a good bactericidal agent. So later on we add the glycerin and peptone. About 40% of glycerin and 1% of peptone is added to it, and the glycerin helps the bactericidal activity of the phenol during its storage at minus 10 degrees Celsius. And the peptone has a crucial role because the peptone maintains it into a viable state. The viral will be kept in a viable state by the peptone, and moreover glycerin also gives viscosity which is easier for administration. This is after it is produced. This preparation can be given as a liquid preparation or can be freeze dried and get administered. So this is how the production from the animals source is done, the smallpox vaccine. So after that a quality test should be performed. The quality test is performed especially against the E. coli as well as an aerobic and anaerobic pathogen. So the quality test should be taken care, and the count of the microbial load should not be more than 500 per ml of the lymph solution. So this is the production of the smallpox vaccine from the animal source. So if you see the production of smallpox from a fertile egg, is that it is an alternative method of the preparation of this vaccine. So the why we prefer vaccine from the fertile egg is that it is easy to cultivate the virus in a egg in a controlled environment, especially the virus grows into the chorioallantoic membrane of the egg, and it is very easy to prepare also. And the major advantage is that there is no contamination issue. So if you see, by scrapping, there is a contamination possible in the animal is there by the bacteria, whereas in this egg there is no contamination is possible, so we can maintain in an aseptic condition. So if you see the production from the fertile eggs, generally the fertile egg is taken, then it is candled for 12 days. After its candling, what we will do is we lamp towards the air sac. The air sac is marked. From the air sac near to the chorioallantoic membrane, a triangle is get marked, as you can see from the figure. So the triangle area is marked so that we have to cut this area. Similarly, in another area near to the air sac, a hole is made for suction purpose. So what we will do is this, first we take the egg and we make a triangle. It is cutted very carefully with the edges of the near to the carbon dump disc. So with the help of a dental mortar, so it is made cutting. Same time a groove is made near to the air sac. After that, what we will do is this cutted triangle area is generally lifted up. So during the lifting of the cell, it should not damage the underlying chorioallantoic membrane. So without damaging the membrane, it is slowly lifted and the sterile solution can be pipetted into the membrane. So which can be easily done with a blunt needle in an aseptic condition. Then a gentle suction is done through the hole we prepare near to the air sac. So because of this suction, the air gets removed. This air is replaced by the fluid content of the egg. So thereby the chorioallantoic membrane where we made a triangle is saw a little depression. So in that we can easily add the saline. So in this new sac, what we will do is we will add the seed culture of the virus into it and we will close it with the same shell triangular portion. So then it is incubated for a particular period, then collected, then purified and used as a smallpox vaccine. So I have told the smallpox vaccine can be given in a liquid form. It can be currently given as in a freeze dried form. So if you see the liquid smallpox vaccine, its potency will be maintained for a year when it is incubated, when it is kept stored at minus 10 degrees Celsius. If the temperature rises little bit above, its stability gets affected, and moreover this vaccine should be protected from light. So if the liquid vaccine reaches to a temperature to 2 to 10 degrees Celsius, if it is stored, then it has a shelf life of only 2 weeks, and 10 to 20 degrees Celsius it is for 1 week. So it is very difficult to administer the patient within this short duration of time. So nowadays we prefer the freeze dried form, because the freeze dried form besides the indefinite shelf life when it is stored at minus 10 degrees Celsius, and it has a shelf life of 1 year when it is stored at 22 degrees Celsius, and 1 month when it is stored at 37 degrees Celsius. So if you see the packaging, the vaccine, smallpox vaccine is available in both single dose container as well as multiple dose container. Generally the liquid preparation is given in single dose container and the freeze dried preparation is given in a multiple dose container. So if you see the dosage regimen, if it is a adult dose, a geriatric dose or with a child dose, so all given prophylactically. If you see the adult dose prophylaxis, first we make a multiple puncture technique, scarification is made, then a drop of smallpox vaccine is administered. So in the adult dose we do the same, like in the geriatric dose also we make a multiple puncture, then we will apply a drop. So in the pediatric dose also what we will do is we do the similar manner, we will do a multiple puncture technique percutaneous and we will apply a single dose of the single drop of the vaccine. So if you see less than 12 months, routine non-emergency vaccination is contraindicated. Similarly, 1 year to 18 years also, routine non-emergency vaccination is also not recommended. So up to 12 months it is contraindicated, and generally up to 18 years it is not recommended. So if you see the storage condition, similar to all vaccine, it is stored in the refrigerator up to 2 to 8 degrees Celsius, and it should be protected from light and it should not be frozen. So I thank you very much for understanding of this vaccine, that is the production of smallpox vaccine. So you continuously listen my lecture on the various aspects of the pharmacy. So thank you very much.